Alright guys, so to pick up where we left off, just the tail end of Lecture 3, the Chemistry of Life. This is the only part that we have left. We're going to look at functioning groups. This is one of those other kind of weird separate topics in chemistry that we kind of throw in there just to help us later on when we look at biological molecules. This idea of functional groups comes from the observation that molecules kind of have repeating patterns. So a functional group is a small regular group of atoms or molecules attached to a larger other molecule. And let me actually give you an example of this on the next slide. Right here, this molecule is a fairly big molecule. This is actually glucose. Oops. This is actually glucose. This is a sugar. And as we'll learn, there's multiple types of sugar, so this is one of our possible sugars. And we can see all over this glucose molecule, there's lots of oxygen bonded to hydrogen. So what we will do in the language of organic chemistry is we'll name this small little repeated bit, these two atoms, and that way we can easily refer to them when we talk about this bigger structure. As it turns out, a lot of these functional groups that we name and that we specifically pull out, they do certain things. They're involved in a lot of different chemical reactions. So they're worth naming. So the OH that we were looking at has a name. It's called a hydroxyl group. It's called a hydroxyl group, and it's simply an oxygen attached to a hydrogen. Keep doing that. There we go. So this is what the hydroxyl group looks like. The R just represents some other big series of molecules or atoms bonded together. A methyl group is a CH3. This is what the methyl group looks like in blue. Alright, I think I have a methyl group. Nope, never mind. Okay, I thought I had a methyl group. Never mind. A thiol group is an SH. An SH, and there's actually a thiol group over here in red. An amino group is a nitrogen bonded to either two hydrogens or three hydrogens. That's why there's that little 3 or 2. It's not 3 halves, it just means 3 or 2. And on this molecule, there's an amino group right there. A phosphate has a phosphorus bonded to 4 oxygens. We're going to see this a lot. Alright. And an acetyl group is a C bonded to an O, bonded to a CH3. Not terribly important for us to know the structure of it right now, but we're going to see it way later in the quarter when we talk about cell respiration. So on this glucose molecule, we have 1, 2, 3, 4, 5 hydroxyls. On this cysteine molecule, cysteine is actually an amino acid, if you guys know what an amino acid is. We have a hydroxyl, a thiol, and an amino group. So a lot of times we'll look at a molecule and we'll say, oh, the hydroxyl bonds with the amino group. So it's a shorthand way of referring to specific parts of a molecule using this terminology. Now you don't have to be able to draw these out, but you don't have to be able to explicitly recognize them. I would never ask you on an exam or quiz, you know, which of these is a thiol group. You'll learn that when you take biological chemistry or organic chemistry. But we should be able to recognize them, right? So given a molecule, we should be able to go, oh, that's a methyl group right there, I think. All right. So that is it for our chemistry. Let's move on and let's talk about biological molecules. So with biological molecules, this is where we're going to start linking together all of our knowledge that we just learned about polar, nonpolar, pH, and things like that, and start looking at the molecules that are found in living things. Biological molecules are different from organic molecules. Remember organic molecules? We made that stupid joke about organic molecules being found in, not being found in Whole Foods or whatever. Remember, an organic molecule is anything that contains carbon. A biological molecule is any molecule that is found in living things. So a molecule can be both organic or biological, or just organic. Most of the time, though, they're both. And I don't know why I have a picture of strawberries here. I think because it looked nice with the purple color scheme I had going. So this lecture is actually separated into 4 parts. And the reason why it's separated into 4 parts is because there are 4 classes of biological molecules. You will recognize these classes, at least for the most part. These classes are carbohydrates, carbs, right? Lipids, this includes fats, proteins, and nucleic acids. That includes DNA and RNA. So we are mostly familiar with these. And we're going to talk about the role of these molecules not only in the cell, but a little bit of their role in nutrition too. So as an example, I know that probably up until this point we thought of protein as something that you need to get a certain amount of. There's lots of protein in steak and meat and things like that. But here we're going to look at not only its role in nutrition, right, but also what it does, what proteins do inside our cells. Now, not almost all, I take that back, 3 of these 4 classes of biological molecules follow this macromolecule structure. So a macromolecule is just a really big molecule. It can be organic, it can be biological. We have a lot of different terms here to describe molecules. And they follow this weird pattern of being built up and being broken down in a manner called monomers and polymers. So let's actually dive right into this. We'll talk about monomers and polymers and macromolecules, and then we'll talk about each of our 4 classes and whether we can consider them a monomer-polymer pair. So now keep in mind that these molecules that we talk about here, these macromolecules, these huge molecules, not only are they truly monstrous, made of thousands to millions of atoms, they make up every living thing. You look at a tree, right? You look at a yeast. You look at a snail. It's made of some combination of these biological molecules. One of the coolest things that I remember after learning this stuff, you know, when I took Bio 160 or the equivalent of it, is I remember walking outside looking at like a bush going, I know what you're made of, right? I could actually like say, oh, I know you're made of this particular carbohydrate, and you're made of this particular lipid. It was kind of cool. Now, just like we have learned before, structure dictates function. A lot of times these molecules, based on their shape, we can kind of tell what they do, and if we change the shape of these molecules, we change what they do. So keep that in mind, especially when we get into lipids. We're going to talk about the different shapes of fats. And those different shapes may contribute to health problems in humans. Okay, let's do this monomer polymer thing. So really quick, what is monomer, or what does poly mean? And if you guessed many, you got it right. Mono means one, poly means many. So a polymer is a long molecule consisting of many similar building blocks, and the monomer is simply the building block. So the analogy I like to use here is the monomer is kind of like the brick, and the polymer is kind of like the brick wall. You attach together a bunch of bricks, you get the brick wall. You attach together a bunch of monomers, you get the polymer. And 3 of these classes of biological molecules strictly follow this monomer-polymer thing. Carbs, proteins, and nucleic acids. Lipids, our last class, kind of sort of do, but it's a little harder to categorize them, as we'll see. So the way that these molecules are structured is actually pretty predictable, these carbs, proteins, nucleic acids. They're structured according to this monomer-polymer thing. So what that does is it makes it really, really easy to build up monomers and polymers and really, really easy to break them down. Let me actually skip past the slide and show you a real example here. Right here, this molecule is glucose. This is a sugar. It's a carb. All right. Here's another sugar, fructose. Notice they have slightly different structures, somewhat similar, but slightly different. And right there at that bend, there is a carbon. There is a carbon. There's a carbon. There's a carbon. There's a carbon. Alright, so we're using our organic chemistry shorthand. And this molecule is made mostly of carbon, hydrogen, and oxygen. We have some hydroxyl groups. Alright, some hydroxyl groups. This right here is a monomer. This right here is another slightly different type of monomer. We link together the two monomers, and here we have a polymer over here on the right. So as soon as we change the structure of these, as soon as we link them together, we also change how they behave. So glucose by itself is kind of sweet tasting. Fructose is very sweet tasting. Sucrose is table sugar. It's what you add to your drinks and to your coffee and things like that. So by arranging our molecules, our biological molecules in this monomer-polymer manner, it makes it really easy for ourselves to build them up and also to break them down. So let's take a look at the reactions where we build them up and break them down. Here we're using just a purple ball to represent a monomer, and a bunch of purple balls linked together to represent a polymer. And here we're taking a pre-existing polymer. Just for the sake of argument, we took just a short polymer. We could have easily just taken another monomer. Alright. And we take an unlinked, free-floating monomer. We take the hydroxyl off one and the hydrogen off the other. Here's the cool thing about monomers and polymers. One will always have a hydroxyl, the other will always have a hydrogen. One will always have a hydroxyl and a fructose, the other will always have a hydrogen. Alright. And we rip off that hydroxyl and that hydrogen, join them together, and we get water out of the deal, right? 2 hydrogens, 1 oxygen. And we create a polar covalent bond right there. And now we have a slightly longer polymer. This chemical reaction, where we do this, it has a couple different names. You can call it either a dehydration reaction or a condensation reaction. Now, why is it called dehydration? Well, maybe it kind of makes sense. Dehydrating, think of it from the point of view of this monomer and this polymer. You're taking away water, you're dehydrating water out of it, right? Condensation will take a little bit more imagination. Now the opposite, hydrolysis, this is where we take a polymer, we add water, and we break it up. You do hydrolysis reactions all the time in your digestive tract. Every time you eat something, you're eating, you're eating billions of biological molecules. You have to do hydrolysis on those biological molecules to digest them. So your digestive tract contains lots of little machines, little proteins, that hydrolyze your food. By the way, the word, or the prefix hydro means water. Lysis actually means to break. So here, notice we're using water to break apart that polymer. Thank goodness something makes a little sense, right? Okay, so here, let's look at this reaction. Here we're taking 2 monomers, right? And we're linking them together using what type of chemical reaction that we just learned? Right? If you answered dehydration, you're good to go. If you answered condensation, you're good to go. The only incorrect answer here is if you were quiet, which I know you were, right? Guilty. It's okay. It'd be kind of creepy if you started like talking to yourself right now. So anyways, this is how plants actually create sugar. They link together glucose, this molecule glucose and fructose, which they make through other processes, and create sucrose. And notice that we get water out of the deal right there. Okay, so let's look at carbs now. We're going to come back and bring in that monomer polymer stuff. Don't worry, we haven't forgotten it. But first let's learn a little bit more about carbs. You'll notice that I have a certain organization here. First I talk about characteristics of carbs, then classification, and then on the next slide, functions. I follow this format for every single biological molecule. Alright, I follow it for lipids, I follow it for nucleic acids, and for proteins. So let's first look at the characteristics of carbs. They are made of carbon, hydrogen, and oxygen in a ratio of 1 to 2 to 1. What does that mean? Well, that means that when you count up all of the carbons, hydrogens, and oxygens in a carbohydrate molecule, For every one carbon, there's going to be 2 hydrogens and 1 oxygen. So looking at this guy down here, this is actually glucose again. This is the same molecule that we saw right here, just kind of skewed a little, kind of flattened. Alright. Remember, there at this apex, there's a carbon, 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 right? Ignore the numbers for now. If you count up all those carbons and hydrogens, I promise it'll be in a ratio of 1 to 2 to 1. In fact, there'll be 6 carbons, 12 hydrogens, and 6 oxygens. Now you notice that all of these carbs that are down here, they form rings, right? They look like a ring. That's really, really common with carbohydrates. They have this ring structure. And if you look at the individual chemical bonds in this ring, there's a lot of carbon to oxygen bonds. Here's another carbon to oxygen. Here's another carbon. It's actually attached to this oxygen over here. So lots of polar carbon to oxygen bonds. And remember, the other word for polar is hydrophilic. Remember, nonpolar was hydrophobic. So carbs are actually really, really hydrophilic. They interact well with water. And if you think about it, here's a carb, right? Here's table sugar right there. What happens when you put sugar in water? It just falls apart. It dissolves, right? Like dissolves like. Polar dissolves polar. Water, polar, dissolves polar carbs. So this is why sugar dissolves in water. When we name carbohydrates, most of their scientific names end in this OSE ending. So glucose, fructose, lactose, sucrose, things like that. You can always tell a carb based on that OSE. So let's look at the classification of carbs. Alright. We can classify them, it's really easy, just according to how many rings there are. If there's just one ring, we call that a monosaccharide. Again, mono, what does that mean? That means one. Saccharide means sugar or sweet. Most carbohydrates are somewhat sweet tasting when we eat them. They're delicious. So this literally means single sugar. Disaccharide means 2 monosaccharides linked together. And how they're linked together is via what kind of reaction? Dehydration, right? Or condensation, whatever you want to call it. A polysaccharide is 3 or more rings linked together, and polysaccharides can get to be thousands of rings. And they're linked together in the same way using dehydration reactions. Now, if I asked you what was the monomer of carbohydrates called, what is the monomer of carbohydrates called, you might have to think about it a little bit, but hopefully you'd be able to answer the monomer of carbohydrates is called a monosaccharide. Alright, the monomer of carbohydrates is a monosaccharide. The polymer of carbohydrates, there's actually kind of 2 of them, is both disaccharides and polysaccharides. Alright. So let's take a look now at their functions. This is where it gets kind of cool. We use carbs for a lot of different things in cells, as it turns out. They do a ton. By far and away, the most common use for them, though, is for short-term energy storage. When I say short-term, I mean like these carbs, they last you maybe 20 minutes during some intense exercise. So you actually do have a store of carbohydrates in your liver and in your muscles, and you'll draw on that store of carbohydrates and burn them in order to generate energy. And if you're really doing intense exercise, you can use those you have enough carbohydrates that'll sustain you for about 20 minutes. And unsurprisingly, different organisms will use different types of carbs, right? So plants use a slightly different set of carbs than animals do. Plants use a carbohydrate called starch. Plants use glucose and sucrose. Animals use a carbohydrate called glycogen. We also use glucose, as it turns out. And we use something called lactose. Maybe you've heard of lactose. Maybe you have a friend who's lactose intolerant. We'll see that. We'll learn that. So that's one of the primary functions of carbs, is just to store energy that you can easily burn and use. One of the other things that carbs do is they form cell walls. So plants have a nice thick, rigid cell wall that keeps them upright. That's why plants are, well, most of them are pretty rigid. That's due to a carbohydrate called cellulose. Notice that OSC ending, right? Cellulose, glucose, lactose. I know we kind of broke our rule with starch and glycogen. Suck it up, you're just going to have to deal with it, okay? We don't have cell walls, we don't have cellulose. We're kind of bendy, we're kind of flexible. Cellulose gives plants rigidity and structure. Yeah, it's not the only molecule that does this, but it's one of them. So when you say knock on wood, right, you're really knocking on cellulose. Alright, bacteria use a slightly different version, slightly different carbohydrate for their cell walls. Bacteria use a molecule called peptidoglycan, right? Good luck remembering how to spell that one. Peptidoglycan. Another thing that we use carbs for is for cell communication. One really good example of this is actually the ABO blood group in humans. So you may know that we have type A blood, type B blood, type AB, or type O, and that's actually simply because of what kind of carbohydrate we have in our red blood cells. We also use carbs in order to help absorb water and to help retain water. So we'll look at an example of a particular carbohydrate called a proteoglycan, another heck of a speller, right? Or another heck of a spelling, I should say. We find proteoglycans a lot everywhere, but we're really going to focus on where they're found in humans, because, you know, we're selfish like that. Okay, so now we are taking a closer look at monosaccharides. Alright, closer look at monosaccharides with some specific examples. So remember, monosaccharides are in this ring structure. The most important monosaccharide that we must remember is glucose. It's glucose. It's the most important monosaccharide in life. And the reason why is it can be transformed into almost any other biological molecule by the cell. Your cell can take a glucose molecule and turn it into vitamins. Your cell can take a glucose molecule and turn it into DNA, can turn it into its cell membrane, can turn it into its mitochondria, if you know what those are. So glucose is kind of amazing. And besides just being able to repurpose glucose for pretty much everything, glucose is a main source of energy for a lot of living things. Most of your daily caloric intake comes in the form of this molecule, comes in the form of glucose. So it would be beneficial, not required beneficial, if you were able to recognize this monosaccharide. So if you can point at this and say, yep, I know that one is glucose, as opposed to one of these other two. Another example of a monosaccharide that we will need to know is ribose. You don't need to know the structure of these other two, just what they do. Ribose is used to form the backbone of RNA. Remember, we looked at our central dogma of molecular biology, DNA to RNA to protein, and we said, well, we kind of don't know what RNA is yet. Here we're just laying the foundation for learning what RNA is later on. So ribose is a monosaccharide that's part of RNA. In fact, if you look at the full name of RNA, ribonucleic acid, RNA, that ribo in the very first part of ribonucleic acid refers to this ribose. Fructose, on the other hand, very sweet tasting, right? It's found in fruits and veggies. It's what makes them sweet, and it's used by plants as an energy storage molecule. We just like fructose, and we like sweet tasting things, so, you know, what benefits plants also benefits us. Now, all these sugars are nutritionally known as simple sugars. In molecular biology and biochemistry, they're known as monosaccharides. In nutrition, they're known as simple sugars. Sometimes it's even shortened further to just sugars. And you can have a bunch of simple sugars or monosaccharides that are 5 carbons in a ring, 6 carbons in a ring, 3, sometimes even 4. So right here we have 1, 2, 3, 4 carbons and an oxygen in this ring, right? Here we have 6 and here we have 6. So we can have a variety of monosaccharides. And what we're looking at here, this is just 3 of the many that are out there. Okay, let's look at disaccharides. Our 2 examples of disaccharides that we're going to learn include lactose and sucrose. So remember, we create disaccharides through what type of chemical reaction? A condensation reaction. Here we're taking 2 monosaccharides and linking them together to get a disaccharide. And as soon as we link together those 2 monosaccharides, we change the name of the molecule. So here we are linking together the monosaccharide glucose, which we just saw, and another monosaccharide called galactose. We didn't see that on the previous slide, but it's just one of those other monosaccharides that are out there. We link these together using that dehydration reaction, and we get a disaccharide called lactose. And lactose is kind of sweet, alright. It's mostly used by female animals as an energy storage and energy transmission molecule. So it's found in milk, right? So that way the baby can get some energy from the mom. Now, what makes some of us lactose intolerant, though? So many humans, in fact most humans, are actually lactose intolerant, which means that our cells cannot by themselves digest lactose. Our cells cannot hydrolyze, do hydrolysis, on this bond in between glucose and galactose. So the vast majority of people on Earth cannot hydrolyze lactose. They can't break it down. And so they are what is called lactose intolerant. Now, here's the thing. Just because you can't break it down doesn't mean that the bacteria in your gut can't. So with lactose intolerance, if you are lactose intolerant and you drink milk or something like that, I mean, it doesn't kill you, right? It just kind of makes you uncomfortable and gassy and maybe a little nauseous. That's because as soon as that lactose sugar makes its way down into your intestines, and to all the bacteria in your intestines, those bacteria go, sweet, lactose, right? I'm going to break it down, I'm going to hydrolyze it. But the way they break it down is not so great. When they break it down, they produce lots and lots of carbon dioxide gas. And so, when you drink lactose, and those bacteria break it down for you, you feel really gassy. So there you go. Now, sucrose, on the other hand, is another disaccharide. And you'll see that it has a slightly different shape. It's still made of glucose, as half of it, but it's made of a fructose on the other half instead of our galactose and lactose. Sucrose is very sweet tasting. It's used by plants as an energy storage molecule, and it's created the same way as any other disaccharide through condensation reactions. That condensation reaction that creates that bond between these two, this bond actually has a special name. There's 2 polar covalent bonds on this side and on this side, and as a whole, both those polar covalent bonds is called a glycosidic bond. So there's a glycosidic bond between glucose and galactose and lactose. There's a glycosidic bond between glucose and fructose in sucrose. So we're going to learn there's slightly different types of chemical bonds in between each one, or each type of biological molecule. Alright, otherwise disaccharides are mostly used for energy storage. It's not like monosaccharides where there's a bunch of different things we can use them for. Here's a couple examples of dietary mono and disaccharides. Monosaccharides include things like fructose, we just saw those, galactose, mannose. Alright. Disaccharides include things like lactose that we just saw, sucrose. This other one, maltose, guess what? It tastes malty, right? Maltose is a disaccharide found in barley, it's found in malty beer, it's found in malted milkshakes, right? You can get malt powder. It's delicious. It's also my favorite drink, Yoo-hoo. I don't know if you guys have ever heard of it. It's that really, really crappy chocolate drink that's kind of chalky, and it tastes like, at least to me, it tastes like childhood. Anyways, looking at polysaccharides now. Looking at polysaccharides. So polysaccharides come in a lot of different varieties. We are going to learn 4 of those varieties. Alright. The first one we are going to look at is called cellulose. Cellulose is a bunch of glucose molecules linked together. This right here is a glucose molecule, glucose molecule, glucose molecule. How do we link together those glucose molecules? Through what chemical reaction? If you guessed dehydration, give yourself a pat on the back. Alright? And this bond in between these glucose molecules, it has a special name. What is the name? It's called a glycosidic bond. So we have a glycosidic bond here, and projecting off into the distance here, right there. Now, both cellulose and its buddy over here, glycogen, are made of glucose. Glycogen is also made of glucose. But notice that the glycosidic bonds are arranged slightly differently. So these glycosidic bonds are kind of at a sharp angle. These are more straight, so glycogen has a different structure. And the alarm bell should be ringing in your head. You're going, different structure, different function, right? So cellulose is actually one of the weird carbohydrates that's insoluble in water, doesn't mix with water. Glycogen does. Alright. Cellulose is found in plant cell walls. It's rigid, it's firm. Glycogen is found in animal muscles and liver. It's not rigid and firm. Cellulose is mostly just for structures, to keep plants together. Glycogen is for short-term energy storage. It's for short-term energy storage. That 20 minutes that it takes to burn through that glycogen through an intense exercise is our short-term energy storage. Alright, so that's cellulose and glycogen. Again, both made of glucose, but slightly different bonds means that they behave totally differently. Really quick, actually, this is kind of cool. And very gruesome, I will warn you. Okay, so if you have a weak constitution, now's the time to hit pause. One of the things that happens when you panic, when you have a panic attack, or when you have, when you get a rush of adrenaline, is you actually break down the glycogen in your livers and muscles really, really quickly so you can get quick, easy energy. So when you have a rush of adrenaline, that triggers what's called the fight or flight response, right? It's when you get ready to punch somebody or run away, right? It's when you're in a fight or when you're getting ready to run away. We experience this all the time during an exam or a quiz, this fight or flight response. And one of the things that our nervous system triggers as part of a fight or flight response is the hydrolysis of glycogen. So we will break down that glycogen, we will hydrolyze it, break it apart into glucose, and then burn the glucose for quick, easy energy. So as a result of an adrenaline rush, we get, well, a rush of energy from that glycogen. And this is what happens. I had a student who was a butcher tell me this, and I confirmed it with a couple others. He said that when you are slaughtering animals, you have to be very careful not to startle them and not to scare them right before they're slaughtered, because it changes the texture of the meat. Let me actually show you a picture of this. Okay. So here we see a muscle cell down here. This is 2 different pictures of different muscle cells, but it still gives us a good idea. Here's a muscle cell before exercise. And this is the very, very inside of the muscle cell. We see some of the organelles, like mitochondria, if you guys know what that is. But what we see a lot of are these little glycogen granules. Here's what a glycogen granule looks like in our muscle cells. Each little black ring here is a glucose monomer, or glucose monosaccharide. In the middle of this rainbow thing is actually a protein core that kind of holds it all together. So here's a muscle cell before exercise, here's a muscle cell after exercise. The mitochondria are kind of squished in this one, but what we can really see is the lack of glycogen granules because we've burned them all. Here's another cell, another image. This is a different cell. I think this is actually a liver cell in this particular picture. Here's the nucleus of the liver cell. Here's a couple mitochondria here, and then you can see those dark little black glycogen granules in there. So anyways, what happens during the fight or flight response is you burn those glycogen granules, alright, and it changes the texture of your muscles. And what is meat if not muscle, right? So you have to be really careful to slaughter those animals humanely so that they don't burn their glycogen and they don't produce a different muscle texture. And as I understand it, the muscle texture that's produced as a result of glycogen depletion is kind of gummy and kind of chewy, so it's not very pleasant. And I think I just turned half of you guys vegetarian. Okay, so let's look at two other polysaccharides, peptidoglycan and starch. When you look at peptidoglycan, it makes your eyes want to like cross, doesn't it? It's so bizarre looking. It's made not of glucose, it's made of a huge variety of monosaccharides and even some things called amino acids thrown in there just for shits and giggles. Okay, one of the really cool things that we do know about peptidoglycan though is that not only does it form bacterial cell walls, many of our antibiotics actually inhibit the synthesis of peptidoglycan. So it inhibits the condensation reactions that create these glycosidic bonds right there. If you stop those condensation reactions, you can't build peptidoglycan, and if you're a bacteria, that means you don't have a cell wall, that means you're dead. So, I think I'm right here. I think vancomycin, if you guys have ever heard of it, vancomycin is a common antibiotic that actually kills bacteria by preventing the creation of peptidoglycan. Okay, starch. Starch is made of, it's going back to glucose again. So this molecule right there, good old friend glucose, right? Linked by glycosidic bonds. Starch is made of just thousands of glucose molecules, and it's mainly used by plants as an energy storage. So, to contrast, cellulose is used by plants, it's for structure. Starch is used by plants, it's for energy storage. Glycogen is used by animals for energy storage. Peptidoglycan is used by bacteria for structure. Alright. Here's, by the way, is what peptidoglycan and what cellulose looks like, kind of on the broader scale. You don't need to know these. It's just kind of cool, and it gives us some context. Here's a bacteria right there. This is actually kind of what E. coli looks like, this rod shape. And if we zoom in on the cell wall, the brown layer right there, the brown fibers that we see, and then the green little balls, both of those are actually peptidoglycan. So that's kind of what a bacterial cell wall looks like. This is what cellulose looks like, right here. Zoomed in, really, really close. As you can see, it forms these long fibers that are quite rigid. Alright, so here's one more function of polysaccharides. Okay. Now we got through the short-term energy storage. We got through the cell walls. Notice all of our examples are listed here for you. Now we're getting into these other two examples, cell communication and water absorption and retention. For cell communication and water absorption and retention, we also use polysaccharides. So let's take a look at, oops, too far. Let's take a look at cell communication now. Right here, here's the surface of a red blood cell. Now, actually, let me see if I can draw. Never mind, I can't draw in here. I kind of can. Okay. Forgive me, really, really crummy drawings. Alright. So, let me do a red blood cell. Right there. Red blood cells are red. Okay. And right in the outer surface of that red blood cell, right along what we call the cell membrane, there are these polysaccharides embedded. So these little blue balls with the sticks, that's the cell membrane, and embedded in the outer layer there's these polysaccharides. Let me draw on the polysaccharide. Kind of stick it out on the surface right there. Hey, that's not bad. Alright, so we have polysaccharides sticking out from the surface of our red blood cells. And these polysaccharides kind of act like dog tags for our cells. They're a method of communication and identification. So you may know that if you are transfused with blood from a person who does not have the same blood type as you, you will mount an immune response. Your immune system will kind of freak out and say, oh, that's not me. Better attack and destroy it. So these polysaccharides are kind of like a little signaling system to our cells and to other people's cells saying, hey, I'm a Liz cell, right? Don't attack me, okay? And as it turns out, humans have 3 major types of polysaccharides on the outer surface of their red blood cells. I'm actually kind of proud of this drawing. Anyways, we have what's called the type A polysaccharide, and when you look at it, when we're looking at this polysaccharide, ignore the green part down here. When you're looking at this polysaccharide, each little gray hexagon here, that's a monosaccharide. A type A polysaccharide will have a particular monosaccharide on the outside. We're not going to say which one, we're just going to say the red monosaccharide. If you're type B blood, you have a blue monosaccharide on the end of your polysaccharide. If you're type O, you don't have any special monosaccharide on the end. You're essentially blank. So your blood type is determined by which monosaccharide or which polysaccharide, or which combination of polysaccharides you have. If you have type A blood, this is the only polysaccharide you have on your red blood cells. If you have type B blood, this is the only one. If you have type O, this is the only one. If you have type AB, you have a combination of both of these on the outer surface of your red blood cells. And it's important, if you ever get a blood transfusion, that if you're type A, you only get blood that's type A or type O. Remember, O is kind of blank to us. If you're type B, you can only get a transfusion from type B or type O. If you're type O, you're kind of screwed. You can only get a transfusion from type O. We'll talk more about this later on when we talk about cell communication, get a little more in depth on this. Alright, here's one really cool example. So polysaccharides are also used as a way to trap water. Remember, carbs are hydrophilic. They interact very well with water. And when you dissolve most carbs in water, it kind of makes them sticky and slippery, right? Think about sugar in water. It's kind of sticky, right? And we particularly use one type of polysaccharide called a proteoglycan in order to trap water. And one thing to note, another word that you may hear for proteoglycan is this term glycoprotein. Alright? I can't make it bigger. This term glycoprotein and proteoglycan are, for most purposes, interchangeable. So if and when you guys take anatomy and physiology, some instructors will use the term proteoglycan, some use glycoprotein. We just gotta know both, okay? And when you look at a proteoglycan, there's 2 parts to it. There's a protein part, hence the proteo, and there's also the carbohydrate part. So if we look at this cartoon up here, the carbohydrate part are these little green balls, right? That's each little green ball is actually a monosaccharide. The protein part is in purple. So proteoglycan is a kind of a combo of both. If you look at a really, really powerful microscopic image of a proteoglycan, it kind of looks like a nightmare centipede. Right? This thing is just creeps me out. And then it's made of a wide variety of monosaccharides. Here's one monosaccharide, here's another one. So we don't have to worry about that here. What we do have to worry about is kind of the take home here. What are proteoglycans for? They're used to trap water. So, I don't know if you've ever done this. If you've ever cut yourself or something and after the bleeding stops, you kind of look into that cut and you see what you're made of and you see kind of this clear gel. Well, that's actually proteoglycans. In fact, almost every bodily secretion that you produce, tears, saliva, right, has at least some proteoglycans. It's kind of sticky. Now, where we have a lot of proteoglycans is in our joints. That's to make our joints nice and slippery so they move past each other, right, and they don't grind. In fact, the cartilage covering the ends of your bones has lots and lots of proteoglycans. And one of the things that can happen as we age is we lose that protective proteoglycan covering, and that results in arthritis. So osteoarthritis, it's one form of arthritis, there's multiple types, is where we actually lose that protective coating of proteoglycans on the ends of our bones, and our bones just grind together and they hurt instead. Alright, that's it for carbs. You made it through. That wasn't so bad, huh? Don't say anything. I know that was bad, but pretend it was good, okay? I hope you guys enjoyed, and thanks for sticking with me.